bacteria. Okay, enumeration is counting. We need to count them. So as I've mentioned, when you need to, only in specific conditions you need to enumerate bacteria. Okay, usually in the food industry, okay, where the level of bacteria in the food will determine whether the food is clean enough to be sold or to be exported. Okay, and in some cases, in clinical settings, for example, in urinary tract infection. So urinary tract infection, the level of the bacteria in the urine will determine whether you are experiencing a urinary tract infection. If the counts that you observe are very, very low, it could just be a skin contamination of the sample. Okay, so these are examples of where enumeration of bacteria in your sample becomes important. So if you looked at some of the video that I've uploaded earlier, pour plating, right, and spread plating. Okay, so these two is related to culture work. You are culturing your bacteria and then enumerating them. So you want to see how many of them can grow. This term is important because there is also another where they look at total bacteria. Okay, so total bacteria uses a different approach where you count all the bacteria in your sample, irregardless of whether you can grow them or not. So irregardless of whether they are culturable, irregardless of whether they are alive or dead. Okay, so there's a very important difference between culturable and total bacteria. So for this, we are focusing on culturable bacteria between pour plating and spread plating. So as I've mentioned, when you're doing culture work, you do not really know the range of the bacteria in your sample. Okay, because if you're working with a new environment, you do not know how many bacteria is in your sample, how many bacteria that can grow that are in your sample. So but from literature review, from previous work, if you read enough journals, then for example, I can take a guess. If I'm working with marine water, seawater, I can guess about 10 to power 2, 10 to power 3 CFU per mil of seawater. I might be able to get them to grow on a marine agar. Okay, but if I'm working with sewage water, then of course I expect more, maybe 10 to the power of 6, 10 to the power of 7. Okay, so the range of bacteria that can grow depends on the type of sample. So if you do not know what is the range, you have to carry out a zero dilution. Okay, zero dilution. You have to dilute your sample. Okay, so if I have a sample here and I dilute it with a diluent, so let's just say I have a 9 ml diluent here and I put 1 ml of sample here. So when I do this, the concentration of the sample is now 1/10, right? Because it is one sample in 9 ml of diluent plus 1 ml of sample. So it is 1/10. So this is 1/10, 10 to the power minus 1, or 1/10 the concentration, the original concentration. Okay, 1/10, 1/10, or 10 to the power of -1. Concentration of the original sample. Okay, this is also can be also mentioned as you have carried out a 10 times dilution. Okay, so 1/10 of a concentration and 10 times concentration. So this you must understand this concept. Are you all okay with this?
Yes.
Yes.
So if that's the case, then if you have 1 ml in 4 ml diluent, what is the concentration of the original sample here?
1/5.
1/5.
Okay. So it's very straightforward. So let's just move on then. So if that is how we do the dilution, then serial dilution is just a series of similar dilution that you are doing in order to get a range of concentration. So if I do 1 ml of sample in 9 ml of diluent, this will give me a -1, -2, -3, -4 concentration. And agree? Everyone
Yes.
follow?
Yes.
Yeah.
I know
Yes, doctor.
you following.
Yeah.
Okay.
Okay.
So let's start with the first rule. Okay. So now you have done this dilution. You've put 1 ml in pour plating. So when I mention one pour plating, I usually will state 1 ml, 1 ml of sample, 1 ml of inoculum. Okay, 1 ml of inoculum in a plate to carry out pour plating. So the first rule of so then you will get a range of counts. Let's just say you got this. Okay, so this means there are 350 colonies on the plate, 40 colonies on the plate, 4 colonies on the plate, and 0. Okay, so the first rule, first guideline for counting CFU per mil is the range between 30 to 300, right? So if 30 to 300, which one is within the guideline?
Second one.
40.
Yes, so very straightforward, 40. So this is 40. So that means this is the plate that you will use. This is the count that you will use to calculate the final CFU per mil. So this is 40. Then you know the dilution factor is 10 to the power 2, right? This is CFU per mil. So this is the final CFU per mil of the original sample. So the first guideline is 330 to 300, okay? So if you have this, okay, what is, so it's the final answer will be, okay? Agree?
Yes.
If you all are not sure, you should ask, because your first test will be based on this. Okay. So what if you have, let's say you have what do you do now?
Count
You have-
ratio.
The average.
Okay, you have two. You have two plates which is within the range. So there are two plates that you can take. Okay, there are two plates here. So you have to take the average. So the average is 300 plus. Why 320? Because we are bringing the count here to this concentration. So if at this concentration is 32, at this concentration it should be 320. So you divide by 2. So the dilution factor is 10 power 1. So it's CFU per mil. Agree?
Yes.
Yes.
Okay.
Second one.
If you're not sure, please ask. So, okay. So you've done this, then you've done the average. Okay. Next, let's just say you have this, then you have this. Okay. So what do you do here?
Count
You have two
ratio.
plates.
You have to make count number. Count ratio.
Ah, yes, yes. Rosmanina. Rosmanina. Correct. She obviously watched the video. Okay. So what is count ratio? This two is within the range. But you realize that this is a little bit high. If this is 1/10 of this concentration, it should be around 30. So the second rule states that the count ratio should be less than 2. No, if the count ratio is more than 2, you have to take the lower dilution. So count ratio. Okay. So how do you measure count ratio? So the count ratio will be
700.
300 divided by 70.
Divided by 2.
So this is more than 2. That means there's something wrong with the dilution here. So we take the lowest dilution. So the answer will be, okay, so that will be 3000 CFU per mil. Okay, so that is the second rule. Okay, agree? Count ratio, okay?
Okay.
Yes,
Okay.
doctor. Doctor, I have a question.
Yeah.
So the count ratio should not be over 2, right?
Yeah.
Can it be under 2? Like 1 point something?
Yeah, if it's 1 point something, what do you do?
You take the average?
Yes, yes, correct.
Okay.
Okay. Next, what if you have okay. So this is obviously a sample that you did not dilute adequately. This is too numerous to count. Too numerous to count. Too numerous to count. So when you manage to count it, it's 5,700. So a normal petri dish is about 56 cm squared, and the maximum theoretical colony per square meter is about 100. So that means the maximum number of CFU that you can see on a plate is 5,600. This is the maximum. Okay. So I'll consider the other rule will be maximum is maximum is 5,600 CFU per plate. So we're talking about plates that are of normal sized. Okay, there are plates that are bigger and plates that are smaller. So you have to adjust yourself according to the surface area of the plate. So this is more than 5,600. So you have your answer will be okay. The dilution factor, okay. Because this is outside the range, you include the term estimated. Okay, so this is how you report your answer. So it's more than 5,600 times 10 power 4 estimated CFU per mil. Okay. Any questions? So if you get this, what should it be then? It is still less than 5,600. So you can report it as 5,200. You times the dilution factor. You have this. Okay. So as long as it doesn't go above the theoretical value, you do not have to add this more than sign.
Doctor, if just now the number was 5,700, right?
Yeah.
Is that possible in real life to get 5,700?
Because sometimes it depends on how you split the plate up.
Oh, same. Okay, same thing. All right.
So sometimes if you calculate, yeah, you can get it.
Okay.
But of course, once the numbers are so high, it is statistically not accurate anymore. You are supposed to aim for this range.
All right.
Any questions until now?
No.
No, doctor.
Okay, so this is what happens when you have lots of counts. So what happens when you don't have counts? All zero. Okay, all zero, don't have counts. So to be sample is really clean. Okay, so what is, how should you report this? Okay, so the smallest count that you can see here would probably be if there is a once colony here. Okay, so if there's a one colony here, it will be this, right? But because you do not even see this, so it must be less than this. Okay, and because it is outside the range, so you report it as this, less than 1 times 10 estimated CFU per mil. So another example, if you didn't do this dilution, but you just did from here onwards. So same approach, you assume, let's just say one colony grew here, times the dilution factor, but even one didn't grow, so you put less than 1, then EST CFU per mil. Okay? Any questions? Hello?
No questions.
No, doctor.
Okay, great. So now that we are here, so this is oh no, there's still one more thing to do. So what if you get let's just say 2200. You report the one nearest to your range. Okay? You times the dilution factor, but this is outside the range still. So you add this. Agree? So this is how you report it. Okay. Any questions? So any questions about CFU guidelines?
Doctor,
Yes?
I have a question about the 5,200 earlier.
Yeah.
It is below the range, right? So do we need to put EST too?
The range is this range.
Oh, you mean this range, not the maximum range?
As long as it's outside this range, you must put an EST.
Okay.
The 5,600 will determine whether you put this in. More than sign.
Okay.
Because the maximum number is only this allowed. Okay?
Okay.
Okay.
Thank you.
Welcome. So we were talking about what if you do not get any counts when you do your spread plating. So if you do a pore plating here, you do a pore plating here, you take your sample, 1 ml sample usually, 1 ml inoculum, okay? Then you get 0 count, okay? 0. Then you report it as so if this is 1 ml of original sample, okay, you get 0 CFU. What is the concentration? What is the CFU per mil here?
Less
Anyone?
than 1? And I'm not sure.
Okay, less than 1. Okay. Everyone agree? So what if you do, when you do spread plating, we usually only spread 0.1 mil of inoculum. Let's just say if you also get 0 CFU growing, so what is the CFU per mil that you can get?
Less than 10.
Very good. So here you can see the difference in the so which method is better for clean waters?
Oh, I think.
Yes, correct. So because the detection limit is able to detect less bacteria. So if you're working with clean waters, then of course you will use a sample, you will use an approach that allows you to detect a lower concentration of bacteria. Okay. So but please take note that the detection limit here is dependent upon this. your inoculum size. So if you have methods where you can increase your inoculum volume, that means you'll be able to have a significantly finer detection limit. Okay, it depends on the inoculum size. So we were talking about pore plating and spread plating. But when it comes to food samples, then you will notice that food samples are not so easy to work with. If you think about spread plating, once you have food particles in your sample, there's no way you can spread the plate well, homogeneously. You will have particles on the plate, and it will disturb the spreading of your bacteria. So for food samples, people usually go for a different approach. They will use a most probable number approach. So what is a most probable number approach? Let's say if you have a sample of water sample here, you just dilute it. Okay, you dilute it. So this is a 10 minus 1. Okay, zero dilution. Then later you find out that the bacteria grows well here, grows well here, and grows well here, but doesn't grow here and here. So from this series, what do you think is the concentration of bacteria in your sample? It should be more than 10 power 3, but less than 10 power 4, right?
Yeah.
Should be somewhere here. Because it grows here, it didn't grow here. So it has to be more than this, but less than this. Okay, so this is most probable number. So most probable number approach works well when you carry out dilution to extinction. So the term here is dilution to extinction, which means you dilute until you find no more bacteria. Then you can get a better MPN number. Okay? Because if you imagine you do this series and it grows all the way here, then there's no way. You cannot say the bacteria is more than 10 power 5. More than 10 power 5 can be anywhere. It can be power 6, power 7, power 8. So as long as you didn't dilute to extinction, you can't really tell the number of bacteria in your sample. That's why dilution to extinction dilution to extinction marks a good MPN, which means if you carry out your MPN dilution and you do not meet 0 tubes at the end, that means your MPN is still not good. Okay, the best MPN is always when you are able to find a range where you see no more positive tubes at the highest dilution. Okay. So this is what you can estimate by doing one single tube. So in MPN, in actual MPN, they'll have 3 tube series, 3 tube MPN, 5 tube and 10 tubes MPN. Okay, 10 tube MPN. So that means you have 3 tubes for 3 tubes. For each dilution, 3 tubes. Okay, and then based on the results here, whether all the 3 tubes grow, all the 3 tubes grow, or only 2 tubes grow, a no tube growth. And from here, you can find out the MPN per mil. Okay. So 3 is the maximum. 3, 2. So the first rule for MPN. Okay. So before we do the standard rules for MPN, are there any questions about this?
No. Doctor, before if we use the 10 tube, but I guess in the table they're stated until 5 tubes only, isn't it?
Yeah?
In the table, in the NPN table, there is
Yeah?
not tube. It means that 5, 4, 3, or 5, 5, 5, isn't it? But
Yeah?
if we use 10 tube, that probability we can get higher chances such as 8 or 7. So it is out of the table. So how we can calculate the NPN?
Oh, the 3 tube MPN requires a 3 tube table. A 5 tube MPN requires a 5 tube table. 10 tube MPN requires a 10 tube table. So naturally, if you have 5 tubes and 10 tubes, the probability of getting a better result is higher because you are covering more tubes, more probabilities here. Okay.
Okay, doctor.
But when you think about it, if you were to do 10 tubes here, 10 tubes here, 10 tubes here, it's a lot of work, very tedious. So not many people will want to do that approach. But I'm just saying that it is available. These kind of tables are available. You can do that. If it is important to you to get good readings, MPN readings, then you can do this. So when you look at once you get this code, you refer to the table. So the table is based on probability and statistics. So that's why they call it most probable number. Okay, it's not definite like spread plating or pore plating. It is a most probable number approach. Okay, but why do we still use it if it's just most probable number? Because you see, it's just broth. You're using broth. So when you have solid samples, when you have food samples with particles and all that, it is easier to work with. Okay, you just have to make sure you blend it, homogenize it properly, and then you distribute it, then you will get better results. Some food samples you cannot use pore plating and spread plating because it just doesn't work. So there's no other way, you have to use MPN. And also when you use MPN, there are also advantages MPN. The other advantage is first advantage of MPN is it is suitable for solid samples. Second advantage is it is broth based. So pore plating and spread plating is based on agar, whereas MPN is based on broth. Broth is somehow a better media to resuscitate stressed organisms. So organisms grow better in a broth environment than solid environment. Okay, so there is an advantage to MPN technique. Another advantage is you can increase the volume of your inoculum to 10 mil or even more. Okay, it depends on how big a tube you want to use. So as I've mentioned earlier when we were talking about detection limit, once you can increase your inoculum, your detection limit becomes better. Okay, so that is also another advantage of MPN. Okay, so I've mentioned that you inoculate in tubes and then you observe for bacterial growth. So how do you observe for bacterial growth? So the easiest way is you look at turbidity. If your broth becomes turbid, That means bacteria has grown in it. Other methods, you can put pH indicator, you can put fermentable sugar. Then if the bacteria ferments the sugar, and the bacteria produces acid, the pH indicator will change, and that will tell you that the bacteria has grown in the tube. You can also have Dirham tubes where you capture the gas formed in your Dirham tubes. These are Dirham tubes. So they will capture the gas formed in the broth. Okay, so the gas is formed by bacteria. That means the fermentation carried out in by the bacteria also produces gas. So these are all indicators of bacterial growth. Okay, so when you use MPN, you are not observing colonies. There are no colony forming units. So you need to have indicators to show bacterial growth. So these are all the indicators that you can use to show bacterial growth. Okay.
Okay, doctor.
Okay. So now we go for the guideline. How do you calculate? So when we have a sample, let's just say we dilute it. Okay, we dilute it. So this is -1, -2. Okay, so we did a 10 times zero dilution before we inoculate into your MPN tubes. So each dilution, we inoculate 1 ml into each tube. Okay, so these tubes have broth, okay, or media that can indicate growth. Then you incubate it, you incubate the tubes overnight and you observe. So let's try. Let's just say you get this result. So the first rule is you look at the tubes that are maximum positive, maximum growth, positive growth, all positive growth. There are 2 tubes with all positive growth. Then you choose the one with the highest dilution here. Okay, so you choose the set with all positive growth at the highest dilution, then 2 successive counts. So the code here, the MPN code is 320. You all have your practical schedule with you? If you refer to your practical schedule, is the value 9.8?
Yes.
Yes.
So you all know where to find it? Okay. So the value is 9.8. The code is 320. You observe the middle set. The middle set is at 1 per thousandth concentration. So your MPN is so we are only concerned with the concentration of the middle set. Understand?
Yes.
Yes.
Any
Yes.
questions? Zulaika?
Yes,
Faham?
faham.
Leon?
Yes, doctor.
Do you understand?
Understand.
Okay. What about Kuganisha?
Yes, doctor.
You okay?
Yep.
Okay. So this is MPN. Okay, this is the first rule. What is the next rule? So what if you get results like this? The first rule still stands. You find the tube, the set with all positive tubes at the highest dilution. So it's still 320. Okay, this one most probably is a dilution error. What happens when you have this?
Add the fourth one to the 3 to 1.
Okay. Yes. So
3-1.
the first rule is take the highest dilution with all positive tubes, then the successive 2 dilutions, but you have one here, so you bring this one here, so that your MPN code is 321. So what is the code for 321? What is the MPN table for 321?
15.
15.
So 15, same concentration. Okay, so that's how you calculate your MPN. Okay, we'll continue. Can we continue? Yes, now. So, what if all 0? If it's all 0, you have to choose the one at the lowest dilution. Look at the middle set. 0, 0, 0. What is the MPN code?
0.3.
So less than 0.3. The middle concentration is 1 per 100. So it is less than 30 MPN. Okay? Agree? Can you all follow? Okay?
Okay.
Okay.
Yes. Okay.
But what if it's all? So when it's all positive, you have no choice. You take the highest dilution. So 333, what is the answer for 333? Anyone?
More than
More than
240.
240.
240. The middle tube is, the middle set is, okay, so the answer will be okay. But if you remember, MPN, the best MPN will be when you have dilution to extinction. when you have results like this, it is not really useful because it can be anywhere. It can be because you still didn't get any negative tubes. So your results is not really very useful. Okay, so dilution to extinction is an important principle for MPN. Okay. So okay. So let's try something strange, different. If I have a sample here, okay, I have a sample here, I have 3 tubes here. So I place 0.1 mil here. Okay. I place 1 mil here. Then I place 10 mil here in each tube. Is this a valid MPN?
I guess not, because we must first we must dilute it into extinction. We must carry the NPN must be carried out by serial dilution before we calculate the NPN.
Is there a dilution here?
The said before, it is on the sample, isn't it? We just take 0.1 milliliter sample, 1 milliliter sample, and 10 sample, and there's no dilution.
So this 0.1, is it the same as this? Yes?
Yep.
This 1 mil, is it the same as this?
Yep.
10 mil, is it the same as this?
Yes.
So is there zero dilution?
Yes.
So it is valid because
Interesting.
there is zero dilution. But I'm showing you this because MPN, you are bound by your middle set, the concentration of your middle set. So it is always possible to increase the volume of your sample. But the problem is here. If you add 0.1 mil into broth, will the bacteria grow well?
Yes.
If you add 1 mil into broth, will the bacteria grow well?
Yep. Kind of.
If you add 10 mil into the same volume of bacteria broth, will
No.
there be enough nutrients?
No.
So that is because MPN requires you to observe growth. If the bacteria cannot grow well, then there's something wrong. So for this one, you have to do a 2 times broth. Only for this set, we'll do a 2 times broth. So we will double the concentration of the broth. So when you add 10 mil of solid volume sample into it, there'll still be enough broth. Understand? There'll still be enough nutrients for the bacteria so that the bacteria grows well.
Yep. But it's a waste of sample lah kan. I mean, like you use a lot of sample. This disadvantage of this way.
Yeah, but that is the disadvantage is very tedious. You use a lot of volume. You use a broth, right? More broth, right?
Yeah.
Broth is, but there's no other way. Spread plating and pour plating will not allow you to do this, especially when you work with samples that are solid. If you blend it and homogenize it, there's no other way, right?
Yep.
So just showing you the alternatives so that you know that if need be, if you have to do it, you can do it this way. Okay, but as your classmate has mentioned, if it is not probably not practical, you know, in terms of cost, in terms of time, logistics. But if you have to do it, then you cannot avoid it. Okay, but I'm just telling you that this is still a valid MPN. You can do dilution so long as your concentration is diluted 10 times like this. Okay, so this is still a valid MPN. So you've done culture, you've enumerated culturable bacteria, we've talked about pour plating, we've talked about spread plating, and we've even done the MPN, right? MPN technique. So pour plating, the first thing that comes to mind is the thermal stress, right? Because you're mixing it with molten agar, there'll definitely be some thermal stress. Then the bacteria grows inside the agar, it'll be harder to work with. Okay, but of course your inoculum is higher, 1 mil, then grow inside, okay, inside the agar. So for spread plating, it grow on top of the agar, so it's easier to work with, but you are limited by the amount of inoculum that you can add in, okay, you can use. So your detection limit is not very good. And then there is also the physical stress where when you spread the when you use the spreader onto the bacteria. So the MPN is different because the MPN uses broth instead of solid. So there's an advantage here. Then the volume also can be much higher, the inoculum volume. So there is also another advantage here. But of course, it is tedious. Okay, but it is good for solid samples. Okay, so next we talk about membrane filtration technique. You can see this. Okay, this is membrane filtration technique. So membrane filtration technique. So basically we are filtering it. There's a filter here, a sterile filter. The filter can be of 0.45 or 0.2 micron. So these are the sizes used, the pore size used that are suitable to capture bacteria. So what we want is we want to capture bacteria. So the bacteria will be trapped on the filter. Okay. Then the whole filtration set can you have glass sets, you have also plastic sets that are autoclavable. Okay. Then you place your water here, your sample, you filter through, then you take the filter and place it on the agar. Okay. Okay. So you have this funnel with a filter here. Okay. So your sample is here. Then you filter it through. Later on top of a petri dish, suitable agar, you will place the filter on top. Okay. Do you place it, if this is top, this is bottom, do you put the top side top on up or down?
Down.
Everyone say down.
Down.
It's the other way, top. Okay? The top side up.
Interesting.
Yeah, because maybe if you're worried that the bacteria will not get the food, then you said, oh, you flip it around. But then you don't have to worry about that because the nutrients will absorb through the filter. So the bacteria will get adequate nutrients. Then the bacteria will grow. Because if you flip it the other way around, even though the bacteria is growing, you can't really see anything. It'll be blocked by the filter. So it has to be the top side up. Okay?
Yep.
Okay,
And
top side.
the
Yeah.
bacteria can grow on the filter also.
Yeah.
Okay.
But the bacteria grows on the filter, but it doesn't use any of the nutrients. There's no nutrients in the filter, but it can be absorbed through the filter.
Yep. Okay.
Then so long as the bacteria grows, it will grow and multiply and make enough of a colony, then you can see, you can observe the growth. But the problem is, the point here is when you place the filter on top of the agar, you must make sure you do not trap any air bubbles. If you trap air bubbles, the air bubbles will prevent diffusion, then the nutrients cannot reach the bacteria. So that is the critical point. So when you place the filter, place it gently, make sure there are no trapped air bubbles. That's all. If there are no trapped air bubbles, the bacteria will grow well. Okay? So you look at this method, then volume of the sample. Can the volume of the sample be high? Yes or no? The volume of the sample for MFT.
Can, I think.
Yes, very good. Can go all the way to 1 liter, 2 liters, no problem. That is why MFT is for clean waters. So if you're working in the mineral water industry, drinking water industry, then you can use MFT to easily filter more than 500 mils. Okay. Why do you need to filter a lot? Because you need good detection limit. Okay. If you filter only 1 mil of the mineral water, will it tell you that whether the water is clean or not clean? When we are drinking easily 2.5 or 3 liters of water every day, do you think a sample of 1 mil can give you good results or reliable results of how clean the water is? No, it's too little. The sample size is too small. Okay, so for drinking water, for clean waters, easily you need sample size of 500 mils and above, so that you really so that you are more confident of the results. So when you have, let's just say you are doing 1 liter of, so 1 liter, if you're doing 1 liter of sample and you get only 1 CFU, so that is 1 CFU per liter. So that is about 0.001 CFU per mil. Okay, if you don't get any counts from here. So that is how good the detection limit of MFT if you use 1 liter sample. Okay, so this is good. No, if you're working in the water industry, you can't be reporting 10 CFU per mil. Okay, you cannot. That is unacceptable. This kind of detection limit is not acceptable in the water and drinking industry. Okay. If you said, oh, well, my water is clean, it's less than 10 CFU per mil, would you dare to drink it? Less than 10 means there can be 9 CFU per mil or 8 CFU per mil. Okay, so MFT is applicable, okay, very useful in the water industry. Okay, we use it often to process our water samples, but do you think it will be good for rivers? Is this good for rivers also?
Maybe not, because there will be too many bacteria.
Yeah, okay. If you have too many bacteria, you have to dilute the samples before doing MFT. So actually it defeats the purpose of having MFT in the first place. So this one the good thing about MFT is probably the low detection limit. Okay, the detection limit is very good. Then usually better for clean waters. Once you use environmental waters like river or other waters for MFT, your first problem you will get is, like your classmate mentioned, the bacterial count will probably be high. Okay, so you need to dilute before you use it. So it kind of defeats the purpose. The second problem is particles. Okay. Because our rivers are not particularly clean, lots of silt, lots of clay, mud. So your membrane is 0.2 to 0.45 micron pore size. So if you use river water with particles inside, if you filter 50 mils, it will clog up easily. Okay, so it will not be easy to work with environmental samples because the filter will clog up easily. Okay, the MFT clogging will be a problem. Okay. Okay. So do you now know the difference between these 4 methods?
Yes.
Okay. So for MFT, you have your membrane filter here. So the best counts will be 20 to 60. 20 to 40, 20 to 60 like that. Okay. Most of the membrane size is about 42 millimeters. Okay, 45 millimeters it is. So 4.5 cm. So if there are too many colonies, they will start to clump together, then you can't get good counts. So usually about 20 to 60. Okay, so we've been talking about culture, okay? Culture. All these 4 methods of enumeration require us to grow them. I've mentioned that there is also another way where we count total bacteria. Okay, total. So direct count approach can be done using hemocytometer. That's like a glass slide where you put the water, your sample on top of the hemocytometer, then you count it through the microscope. So, but as is written here, it is difficult because bacteria has no color, natural color. So it's very hard to observe bacteria if you do not stain the sample. Okay, it's hard to count. Now we use direct counting through epifluorescence microscopy. So basically the setup is the same as your membrane filtration technique, except that the filter used is usually smaller is about 25 millimeters. So the filter used is 25 millimeters and it is black in color. Okay. Why is it black? Because black membranes will reduce the background light when you're viewing it under epifluorescence. So direct counting with epifluorescence microscope, what we do is the setup is the same as membrane filtration, except we use a 25 millimeter 0.2 micron pore size. We use a black membrane filter. Okay, because we are using epifluorescence microscope. Okay. So what we do is you usually put your sample here with a stain. You will stain your sample with a fluorochrome. Okay, a fluorochrome. The fluorochrome we usually use is Duppy. This one will stain bacterial cells in blue or acridine orange. This one will stain the bacterial cells orange or green. Okay, depending on the So usually Duppy. It has to be black because we need to reduce the background light. So if this is your membrane with your bacteria here, okay, we have already stained the bacteria with fluorochrome, Duppy for example. So the epifluorescence microscope has a high intensity light, usually from mercury lamp. So the high intensity light will strike the slides. Okay. So when the high intensity light strikes the bacterial cells, the fluorochrome that binds to the DNA of the bacteria fluoresces. Okay. It fluoresces. So you can see the bacteria will start lighting up. Okay. So when it lights up, the lights go up to your eyepiece. Okay. Then this is the color that you observe. That is why you need to use black filters. If not, the fluorescence of the light will be it'll be hard to see the fluorescence of the light if the background is not dark. Okay, so this is the basis of a epifluorescence microscope, where high intensity light will activate the fluorochrome in the bacteria. Then the bacteria will fluoresce and you can see the color. Then from the observation, you can easily count the number of bacteria in your sample. Okay, so from here you can count the number of bacteria in your sample, then you can calculate back to the number of bacteria in the original sample. Okay, so you can see here that for this direct counting, there is no culture involved. Okay, so the bacteria that you see is not all of them will be able to grow on plates. So the term we use for direct counting is the number of bacteria that you get from direct counting is total bacteria. So as opposed to culturable bacteria, which is in the unit CFU per mil.